pleasure to introduce Aryeh. You can start sharing your screen already. Aryeh did his PhD at the Washington University in St. Louis, and he was working on macroevolutionary innovations and adaptive radiations on anomalous lizards. He's now a Peter Bach postdoctoral fellow at the Smithsonian National Museum of Natural History. Guess he's working on similar topics on evolutionary genomics. And today, he'll be talking about the genomic base of parallel adaptive radiation. On to you, Aryeh.
Can everyone hear me okay?
Yeah.
It's all good. Everyone can see? Okay, brilliant. Well, thanks so much for that very kind introduction. And thank you so much to the wonderful organizing committee for inviting me to speak today and for hosting this fantastic seminar series. I feel let's jump right into it. So today's talk takes part in the Caribbean, but more specifically in the western part of the Caribbean. Here we are. We're in the Greater Antilles, just to put a place to the name. So these are the major arc islands of the Greater Antilles. We have places like Cuba and Hispaniola, Puerto Rico, Jamaica, and some of these smaller islands in the Bahamas. Now, this photograph is from my first time doing fieldwork in the Caribbean. We're way out on the eastern edge of the Puerto Rican bank. This is in the Virgin Islands. And if you were to go out, choose this part of the world, way out in the Puerto Rican bank, you would see some pretty neat reptiles. Now, the Caribbean, of course, is a wonderful place to be a biologist. It's, of course, a wonderful place to be a herpetologist because you would go out and you would see lizards that look a bit like this. Now, this individual is perhaps a bit drab, but this is what we call an anoles lizard. More colloquially, we just call them anoles. A few things about anoles. They are largely arboreal or semi-arboreal lizards, and they have a really, really large distribution. They're found throughout the neotropics, but they go as far north as the Smokies, that's way up in North Carolina, and as far south as the tip of the Brazilian Amazon. They've convergently evolved these sticky digital adhesive toe pads along with their gecko and skink cousins. And we think these play a pretty central role in their arboreal ways of life. They also have these extendable throat fans. So for folks who are interested in speciation, anoles are, of course, a group of interest. And now on that same topic, anoles are one of the most speciose vertebrate genera on Earth. There's about 435 different species of anoles today. So there's quite a large group. I should stop talking about them. I should just show you what they look like. This is Anoles valenciani. This is a Jamaican species. This is a kestrus. This is an animal from Cuba. Here's another cute animal, Anolis barbatus. In fact, anoles come in many, many different shapes and sizes, but in the Caribbean, they really come in 6 primary flavors or forms, and we call these ecomorphs, a familiar term to many of us here. In the Caribbean, ecomorphs are generally distinguished phenotypically and ecologically from each other by the partitioning of microhabitats. So for example, crown giants are the largest anoles on top here. They live in the canopies of trees. These twig anoles, these light blue ones, or Carolina blue ones way on the edge, are really small and skinny. They live on twigs. Now, the thing about anoles here is that they've gone through a process which we call adaptive radiation. This is the process where a single ancestral phylogenetic lineage diversifies into a suite of phenotypically disparate species, each adapted to its own different ecological niche. Now, if we come back to the Greater Antilles here, the cool thing is that if you go to any of these major arc islands, even some parts of the Bahamas, you'll see anoles communities that look very, very similar, both in composition and phenotypically. Well, the neat thing about these communities is that different ecomorphs on the same island are more closely related to each other than identical ecomorphs across different islands. This is what we call, of course, convergent evolution, right? Anole communities across the Greater Antilles have arisen entirely independently. Well, here's what that looks like in practice. Anoles have basically overcome this great historical contingency in their history, where we kind of see the power of natural selection to supersede historical inertia itself. So we reveal this sort of striking predictability to their speciation. On the top left here, this is Anolis garmani. This is a crown giant anole from Jamaica. On the top right here, this is our friend Anolis cuvieri. This is a crown giant anole from Puerto Rico. So in the Caribbean, we have these really nice parallel adaptive radiations, and we see really great convergence in both community structure, so in these assemblages on the different islands, and also in the individual phenotypic adaptations of the animals within those communities. So in a very Gouldian way, and sort of taking off of wonderful life, we might ask, is adaptive radiation hierarchically predictive if we see this scaling cross-biological convergence? I think in 2026, it's appropriate to answer this question now at the molecular level, right? Is phenotypic convergence in anoles mirrored at the molecular scale? Well, we might imagine two ways to go about this. We might see that perhaps there's a distinct genetic basis of phenotypic convergence. So perhaps that's different mutations in different genes in the same ecomorphs. Alternatively, we might see a shared genetic basis of phenotypic convergence. So perhaps that's something like the same mutations at the same sites in the same genes. Now, there's a whole lot of anoles, and this is their distribution, right? All the way up to the southeast of the United States, all the way down through most of Central America, basically, where it's not cold enough. So we spent about the past 15 years gathering tissues from these animals, either out in the field or in museum collections. And what we ended up with was a short read genome for about 99% of all anoles. So that's 436 different genomes since about 2022. Now, each anole genome is around 2 to 2.3 gigabases compared to ours, right? We're around 3.2. So that's 31 and a half terabases worth of data, just to put into sort of scale how much data that is. If you took each one of those base pairs and you just line it up to one another, that's about the distance from New York to Tokyo. So this is, I think, no surprise for folks who work in comparative genomics. It's a lot of data. I just want to emphasize that what we're doing here is sort of trying to build an anolis genome bank so we can answer different questions in speciation and adaptive radiation and comparative biology. And of course, no one works alone. And I have a wonderful group of colleagues at Rutgers Camden, at WashU, at the Smithsonian doing this work. So we're led here by Anthony Geneva, who's now an associate professor at Rutgers. And I have wonderful colleagues, postdocs and graduate students who are working on the reference genomes that were really critical to this project. So anoles have about 19,000 single copy protein coding genes, and we capture about 80% of that in our sampling here. So there's an average of about 9,500 single copy orthologs per species. What you're looking at in this plot is each bar is an individual species. The height of the bar corresponds to the proportion of the protein coding genome that we captured. The color of the bar corresponds to the subgenus of interest here. So there's a number of different subgenuses of subgenera of greater Antillean anoles. So it's great that we have all this data, but we really want to ask just a very simple question straight off the bat, right? What is the evolutionary history of anoles? How are these things related to each other? When we slam all these data into a coalescent analysis, what we see is a really brilliant pattern like this. So we see this really, really beautiful monophyly of greater Antillean anoles, this great rainbow clustering of the different subgenera. So what we have here is our anole tree of life. Now, I want to point out a few things. The first thing we're able to do with this genome scale data is resolve a lot of outstanding recalcitrant nodes. So this is an example where genome scale data actually are quite useful in resolving some of these deeper nodes in the anole tree of life where we've had previously, I've had many issues with perhaps smaller data sets and less comprehensive taxon sampling. Now, a familiar sight to many, I think, can be seen through here on the top left. So what we're looking at is a subgenus level tree of the anoles. And I want you to look at the nodes that are numbered 1 through 5, and you can see how close those are to the root and how short the branch lengths are of the proximity of that short proximity to the root here. Right, this tells us that there's been a lot of incomplete lineage sorting in the early history of greater Antillean anoles, which is signified by those really short internal branches. So we think that these major adaptive radiations in the Caribbean probably arose relatively quickly, probably in the ballpark of about 1 million years or so. We can also infer things about the biogeographic history of anoles. So here on the left here, we have Dactyloa. This is the earliest diverging clade of anoles. These arose on the mainland of South America about 50 million years or so. Anoles then dispersed out to the Caribbean and colonized Greater Antilles, which where I see these really beautiful replicated adaptive radiations that I was just talking about. And then around 30 million years ago, they came back to the mainland and colonized most of Central America. If we zoom in on the Greater Antillean anoles here, so what we're doing is we're taking that big anole tree and we're just focusing on the animals that live in the Greater Antilles. We have a slightly reduced tree, and we have our 6 ecomorphs. And just to sort of tell us where we are again, we're in these major arc islands. And we plot the distribution of each of the ecomorphs across this tree. What we see is a distribution like this, right? This is very different than our subgenus rainbow coloration. I'm just going to bury the lead a little bit here and show you this from a different way. Here are some really lovely illustrations of the ecomorphs of Greater Antillean anoles. So these are different species of the same ecomorph. And if we plot the species tree next to ecomorph, another way to visualize this is just look at the phenotypic convergence spanning out to each of these different ecomorphs. So we can see repeated origins of each of these 6 ecomorphs across the Greater Antilles. So genome scale data really does a great job here to affirm these long-standing hypotheses in our understanding of anole evolution and their very repetitive nature. Okay, very neat. Now we have our newly assembled anole tree of life, and we can begin to look at molecular evolution across and within each of these radiations. So the first thing we want to do is basically do this large genome-wide screen for positive selection, perhaps. We say, what's going on in under the hood for these different biological processes that might be under selection. We take each codon alignment and we just are measuring the DNDS rates across time. We want to put together a table and we just want to say, okay, well, this gene's under selection in this subgenus, and this gene's under selection in this subgenus. And then from this, we can assay, well, perhaps forebrain development is enriched in the Jamaican anoles, while RNA folding is enriched in some of the Hispaniolan anoles and things like this sort. And we can visualize this in a heat map that looks like this. So the most important thing here is that redder tiles signify greater enrichment for a particular biological process. When we do that, we get a distribution that looks like this. So we see significant enrichment in these broad gene ontology processes, things like cellular processes and localization, but also development. And what we really want to do is zoom in on the developmental functions here because these are sort of our primary phenotype of interest in this system. So when we look at the Go terms under development, we see that each radiation has been pervasively and recurrently shaped by positive selection on processes regarding anatomical structure development. This is no surprise, right? Anoles are pretty old. They're about 50 million years old, and we're assaying all this positive selection across the genome. Let's look at this from a slightly different angle. Now, note that we assay positive selection. We're looking at selection that's taking place on specific branches across different points in time. And we have the precise branches in which these are taking place. So what we can do is we can count up the instances of parallel selection. That's instances where we have selection on the same gene in two different subclades, and instances of idiosyncratic or lineage-specific selection. And we can just tabulate these. And we can get this frequency distribution of parallel versus lineage-specific selection across the anole tree of life for many different genes. And this is basically what that distribution looks like, right? There's a lot more lineage-specific selection than parallel selection. Now, what we want to do, again, in a very Gouldian manner, since we've identified these various instances of idiosyncratic and repeated selection across these all radiations, is ask, is there a genome-wide excess of parallel positive selection? That is the selection we observe across the genome exceeding that expected by random chance alone? We expect many loci to be under parallel selection solely owing to random chance alone. It's a lot of time. There's a lot of different protein coding genes. Well, here's what some sort of model for that would look like. Now, recall that we observed a very specific set of genes to have experienced parallel positive selection. So they have names, BMP2, TBX3, these sorts of things. Now, if this observed set is non-random, we would expect that randomly selected genes would be under parallel positive selection significantly less frequently. Well, we can sort of test this using bingo as a model. Now, we might imagine that we have 15,000 different ping pong balls. And on each of these ping pong balls, we are writing the names of our protein coding genes. So something like SOX9 and WNT1 and TBX3. And we're throwing all of our ping pong balls into our bingo machine, and we're going to randomly draw them. And we can estimate a null distribution by randomly shuffling these ping pong balls and reassigning them to different clades. So after reassigning these at random, we can recalculate the total genes to be observed under parallel positive selection. There might be two outcomes here. The first is a model of excessive parallel positive selection, where that dashed line, that's our observed degree of parallel positive selection, is significantly greater than the null distribution. That's that solid big bar right there. Alternatively, we might have a random chance model, right? There's no difference between a random draw of genes and the genes we observe to be under parallel positive selection. Well, here's what we see. We can split them up by internal and terminal branches. And what we find is that in each instance, the observed degree of parallel positive selection is significantly greater than the null distribution, telling us that acribian anoles exhibit a genome-wide excess of parallel positive selection. It's pretty neat. Now, I think if we're interested in understanding the molecular basis of convergent adaptation, we need to shift away from this genome scale or genome-wide approach and really go down to a much more microscopic level to a site level resolution. Now, if we're studying diseases, for example, we might use something like GWAS. This is the common approach in PopGen, where we have a focal phenotype and a case control study. So we have folks perhaps with Alzheimer's and folks who are not suffering from Alzheimer's. And we're looking for linked mutations that are associated with the presence of that focal phenotype. And we're looking for something like an outlier SNP on a chromosome. In macroevolution, we have sort of a similar idea, where perhaps we're looking for a convergent phenotype across different species, and we're looking for focal substitutions on a protein that best predict the distribution of that convergent phenotype, and we're trying to find outliers among the different protein coding genes in the genome. I'm going to give you an example of this work here in some of my favorite anoles. These are the twig anoles. This is Anolis occultus, a Puerto Rican species. I was fortunate enough to see this species in the wild a few years ago. Here's what it looks like. You find them out in night. They are really brilliant animals. Just to give you an idea of how weird these anoles are, put them in your hand. They are really, really small. Now, I think they are particularly small when you compare them to animals that are just a few meters away in the forest. This is a crown giant anole in that exact same plot of land. Okay, so there's a real size disparity in anole communities. Now, what we want to do is scan greater Antillean anoles for non-synonymous mutations that are exclusive to convergent twig anole lineages. We might imagine that across a given amino acid alignment, we have a distribution that looks something like this, so really strong sequence conservation, perhaps with variation in the presence of a phenylalanine, but it's not restricted to twig anoles, for example. This is the signal that we're looking for. We're looking for residue-wise parallel molecular evolution where it's specific to twig anoles. So there, that phenylalanine is only present in twig anoles, for example. Well, we can do this using machine learning pretty well. And we take a given amino acid alignment, we can just one-hot encode it, and we're looking for these trait positive, trait negative contrast pairs across the anole tree of life. And the most important thing to look for here is we're looking for sites which best predict the focal convergent phenotype. And we can rank those loci by what's called the group sparsity score. The most important thing about the group sparsity score is the higher it is, the better predictive value it is for convergence. The lower it is, the worse predictive it is for convergence, basically. Well, here's what we see. If you look all the way up there, it seems like there's quite an outlier, isn't it? That is MSX2. Let me tell you a bit about this gene. This is a big transcription factor, so there's a big homeodomain or DNA binding domain right in the middle of it, and it's pretty conserved across the metazoan tree of life, and we know it plays a variety of roles in the identity and proliferation and migration of different cell types. Here's a good quote here. The main action of MSX genes in both normal and sonic hedgehog knockout limb development is to control cell death in the mesenchyme. If we take some single cell data from embryonic human hind limb, you can see that it's really specific for that distal mesenchyme in developing vertebrate embryos. So it's really specific to that distal part of the hind limb. Well, if we look at the distribution of twig anoles and we're looking across the alignment of MSX2 for this transcription factor, what we see is that there's one site in particular where twig anoles, which are colored in brown or orange here, all have this alanine. So you see this transition from a threonine to an alanine that has evolved at least 3 times in greater Antillean anoles. In fact, it's present in 11 of the 13 twig anole species. Only 2 species that do not have it. One is pictured here, Anolis darlingtoni. The other is Anolis valenciani. These are the largest twig anoles. So we think that this locus has something to do with either limb development or head development. We're not quite sure. We do know that what it's doing is replacing a really polar threonine with a non-polar alanine way on the C terminus of this protein. So this is resulting from this loss of threonine's hydroxyl group. So in sum total here, I think the speciation of greater Antillean anoles tells a pretty interesting story about the historical nature of evolution. I think there's really 3 key findings, which I've tried to present on here, would hit on this. So the first is that anoles have this really strong history of phenotypic convergence. We've known about this for several decades now, but now we're just confirming it through genome scale data. We also see this recurrent nature of evolution through positive selection on development and specifically developmental processes directed to the anatomical feature formation processes. At the genome-wide scale, in these replicate radiations, we see that parallel positive selection has occurred significantly more frequently than random chance alone. And also we see a pretty good example here of really ecomorph congruent parallel amino acid level evolution in a transcription factor that's pretty central to the vertebrate limb morphogenesis program. So anoles tell a nice story where we see that evolutionary history is characterized less by lottery, I think, and perhaps more about repeatability. So repeatability at ecological and phenotypic scales. And I think as we're getting to understand a bit better now, more mirrored by repeatability across molecular scales as well. So with that, I'd like to thank many of the different folks involved with this project and thank the wonderful organizing committee again for giving me the opportunity to speak.
Wonderful. Thanks, Aryeh, for such an interesting talk with pretty pictures. It's really nice to see all those lizards. So we have time for some questions. Feel free to unmute yourself or write it in the chat. Maybe I have a very naive question, a question about them. So maybe you said this and I missed it, but how much do you find a lot of hybrids between these different ecomorphs, or what's the degree of hybridization?
Good question. So we actually find no cross-ecomorph introgression. We find some introgression very, very early in the history of some of these radiations. I would say that research kind of is in its infancy. It's only been studied just a bit. But in general, there's no cross-ecomorph introgression among extant populations. We only find some hybridization among the same ecomorph populations, which are very recently diverged species. So things like some of these green anoles in the Bahamas, for example.
Mm-hmm. Thanks.
I have a question. If that's all right. Yeah. Thanks, Aryeh. That was awesome. I was wondering, is there substitution rate variation in Anolis, or is it something not many people have looked at? Or
Yeah, I think not a lot of folks have looked at it, I would say. I think anoles as a field in general, we're sort of still catching up to some of these other groups who study adaptive radiation, like the cichlid folks and many of these other groups. We're sort of still building up our repertoire of reference genomes, but I think it's definitely an area of research that we should look into.
cool. Thanks.
We have some questions in the chat. So firstly, how do we know if these mutations are independent or introgression or reuse of ancestral variants?
Yeah, so in many cases, we know that just from biogeography that they're arising from de novo variation, which is sort of a nice thing about some of these radiations. In some cases, it's less clear. So we definitely have an example of like hemiplasia, where there seems to be a fourth origin, for example, of this MXX2 mutation. We actually know it's not the case, and it probably arose from ancestral polymorphism in one of these populations. So I would say it's sort of a case-by-case basis, but the distribution of anoles lends itself to, I would say, understanding if it's de novo or some sort of ancestral polymorphism.
Thanks. So we have one more question in the chat. How did you account for gene duplication when doing your analysis if you needed to?
Yeah, so we're only looking at single copy genes in all these analyses, but I think looking at gene duplication is very interesting within itself. But here we're accounting for that.
Mm-hmm. There's another one in the chat from Sahra. Did you solve the structure of MSX2 from each ecotype and align them structurally?
So that's an AlphaFold predicted model. I mean, the tail end of the protein is not, I would say, really predicted that well by AlphaFold. I think because it's kind of a weird evolutionary homologue, there's just not a lot of data for it. I mean, optimally, we would crystallize it. I think it's just nice to put mutations like spatially on a protein sometimes. But that's actually the human ortholog. That's actually not the anolis predicted structure.
So Roger has a question in the chat. How do you think the evolution of divergent ecomorphs relates to the evolution of Aryeh? Does it rely on within island allopatry?
Sorry, do you say reproductive isolation RI? Is that?
Exactly. I guess, yeah.
Yeah, so I mean, I think the classical model of anolis evolution is this sort of character displacement model. where you have, anoles in a community where there's really strong selection and syntopy for character displacement of these different ecomorphological functions. I think that this data set is somewhat capable of answering some of those questions. The hard thing about anoles, which is sort of a double-edged sword, is that the trait distribution is extremely labile. So if you're interested in convergence, that's, I think, a real strength. If you're interested in reconstructing ancestral states, that's a real bummer, I would say. So it's very hard to estimate, I would say, the precise stepwise change of community assembly to understand sequence of RI and things like that. But we know from just experimental data that there is complete RI between these different ecomorphs, for example.
I have a question. I just wanted to ask, thank you for the great talk, first of all. Whether you have a sense of how this degree of like parallel genetic evolution relates to other adaptive radiations, and like what, perhaps if anoles are kind of extreme, if you could speculate just really on what kind of makes them special in that respect.
Yeah, I think it's a great question. It's something we're all trying to think about. Sometimes it's hard to compare across adaptive radiations, but the thing that strikes me as most different about anoles, I would say, is perhaps that one, they're pretty old. So, they're not like cichlids or, geospiza or something like that, where they're definitely pretty old. Two, I think that it's unclear how much they're shaped by introgression. We know that cichlids and finches and many Drosophila even are very shaped by historical introgression or even maybe some sort of concurrent introgression. Anoles perhaps seem to not be that way. So I would argue that the parallel evolution of anoles, maybe there's an angle that this is a group of adaptive radiations that is shaped less by, ancestral polymorphism and more by, de novo variation, for example. But I think it's sort of, we're still figuring that out precisely.
Amazing, thank you.
Cool. Thanks very much, Aryeh. So now we can move on to our next speaker, Dr. Rishi De-Kayne. So Rishi did his PhD at the University of Bern and the EOWAG Institute in Switzerland, looking at the genomic basis of adaptation and speciation in the alpine whitefish adaptive radiation. Rishi is currently a scientist at Arcadia Science, where he works on leveraging evolutionary information to improve our ability to find and leverage useful molecules. And that will be the subject of his talk. So take it away, Rishi.
Thanks so much. And thanks for having me. Thanks for joining. And also thank you for me maybe giving a bit of an out there talk and being open to this. I know that I've worked with many of you and I've admired many of your, papers and projects in the past. So this is kind of a fun example of maybe how we're using a lot of this research base and a lot of these insights in an industry setting. And so, I just wanted to start by saying my past work kind of rests very heavily on the topics that all of you continue to study, and that is asking questions about what might be the interspecific drivers of or constrainers of speciation. What are the actual processes that underpin rapid diversification? And we just had a really nice example of that in the anoles case. Can we tease apart these processes that drive adaptation and speciation? When are they the same processes? When are they different? And how can we better understand extreme traits or, traits that seem to be outside of the range that we typically observe? And obviously these are usually very charismatic, but they also often serve as kind of useful examples. And my current job here at Arcadia is trying to take all of these skills and take this body of work and try and integrate it with what we know about developing kind of useful insights, I would say, in like an industrial setting. And really the way we're doing that is thinking a little bit differently about how we can use these kind of fundamental biological insights. And that involves using evolution as a backbone for biological research. So that's the reason why I work here at a kind of biotech company, which is not super typical. Trying to integrate foundational work with also applied work and kind of building this iteration loop. So doing full loop research where we can look for interesting things, find interesting things, test them in the lab, and kind of use that to develop our tooling. And also this substantial focus on open science. As a company at Arcadia, we try and do everything openly. So all of our pubs, which are at the Stacks, which you should go and check out if you're interested in some of the projects we're doing, they all have reproducible code. A lot of our tooling is open and will be open. And this is something that kind of drives the way in which we do work too. So we typically package projects, slightly smaller packages, I would say, than outside of industry or outside of our immediate kind of reference frame. And that allows us to publish things that are maybe useful internally and useful to people externally, but don't have to be these kind of huge comprehensive stories, which for me at least has been a really fun and exciting way to work. And what we're doing on platform, which is the team I work on specifically, is leveraging computational tools to actually interrogate biology in new ways. So that typically spans integrating across a few topics. So one of those would be machine learning approaches, protein biology, and evolutionary biology. And together, these kind of allow us to span quite a lot of different topics in a way that I think is, to me at least, very interesting, and I think maybe also to many of you. And I've just given 3 examples of some pubs that have come out of our team in the last year. One of them on the left is about looking at reduced representation approaches or dimensionality reduction approaches that are interpretable. So a common pushback against things like UMAP is like, well, I've reduced dimensionality in my data set, but what do the points actually mean? What do the axes mean? And we're developing machine learning approaches that can help us actually dig into those black box models and understand those axes of variation a bit better. Another example here in the middle is to do with protein language models. So these models are being increasingly used, especially in things like therapeutics development. But the reality is we don't have a great understanding of how they work and why they work. And so we've put a lot of effort into trying to understand what components of those models that try and predict residues based on the sequences that are in their training sets are actually contingent on evolution or learn evolution. And learning that is going to be really important moving forward because we're starting to apply these models to a broader and broader set of taxa for which, we know that the training data for those taxas may be a bit more sparse. And on the right here is one of my pubs, and that is trying to look a little bit differently or more integratively about how we actually look for interesting molecules out there and whether or not we can build in evolution guided or explicitly evolution integrated approaches to do that. And that's what I'm going to be talking about today. And the reason for this is that, as many of you know, and I think this is really fun talking to evolutionary biologists, organisms are super weird and they've come up with so many solutions to many different problems. And the diversity across the tree of life is incredible. And the reality is that there are many more interesting and useful innovations across the tree of life than we will ever find, probably, right? So we're kind of constrained by the fact there's just so much out there and we basically don't really know where to look. And historically, people have looked for these things in very tedious ways. And it's kind of remarkable, really, that we have so many drugs today that rely on finding these interesting molecules. This is just an example from Akira Endo. And I'm just want to read this because it's so absurd when you think about it in 2026 for like a typical way of working. So Endo's team were culturing these broths with thousands of fungal species, trying to look for some that showed the inhibitory activity that they were looking for. And they basically ran this iterative approach. They were sampling isolates from all sorts of places, including, in this example, a rice sample collected from a grain shop in Kyoto. This is in the 70s. And what he was saying is that they find that these culture broths develop these molds and that it took 1 year to isolate the active principles from the culture broth. And if you think about that from like a drug development pipeline, you having to culture thousands of unknown strains of fungi to then extract a single isolate over a span of a year that you basically don't even know what you're looking at is obviously not the way we want to go about doing this now. And having more scalable approaches is something we're striving for, given that we have so much more data. So one of the drugs that Endo helped develop is lovastatin, which is this drug that helps decrease bad cholesterol levels. It's a very common drug. And this is extracted from a filamentous fungi species. And drugs like this, we can now understand the structure of these proteins. We can understand the evolution of these proteins. And so trying to integrate the sort of historical aim of discovering drugs from across the tree of life with this like insane data sort of abundance that we have is something that I think is a really exciting avenue. And ideally, this process would look something like this. We can develop pipelines and approaches to find these useful molecules. We can apply them to find interesting things across increasingly non-model taxa. And then ideally, we validate those findings, and we can do that both in silico and experimentally. So the benefit now is we have pretty good in silico approaches where we can screen molecules for their binding ability. But as we do here at Arcadia, we also really value being able to do that experimentally and to actually see, like, when we express these proteins, what is their function, in reality. And the question is, like, if this is such a great approach to doing it, aren't there tons of people already looking across the tree of life to do that? And the answer is yes, there are a bunch of companies doing this. Here are three, just for an example. And these companies are compiling massive data sets. And I'm just going to give you an example of Basecamp research, for example, the one I gave on the right. Here you can see the UniRef protein database here on the far left. And Basecamp research are assembling these amazing, extremely data-rich data sets. So these are protein sequences in billions, and you can see these are orders of magnitude bigger than other data sets that we have available. And these huge data sets also attract a lot of publicity. So you'll see these headlines like, have Basecamp research really discovered 1 million new species, which I'm sure is like very eye roll triggering to a lot of people in this seminar. It was to me at least. And you all see these headlines saying, well, they're going to apply AI and they're going to put all of this data into these black box models, and it's going to spit out all of these drugs, and we're kind of sorted, right? They've just found all this data. And then you kind of dig into a little bit, and what you see is that you'll see caveats like this, like they contain no public data. So nothing from public data sets, things like UniRef, UniProt are in these data sets, but that goes both ways, right? So these companies are assembling essentially paywalled models, and that's their business model. And they are identifying essentially as data companies. They want to leverage the data that they collect. They want to assign labels to their data, and they hope that by doing that, they can build models that will elucidate something that we've been unable to kind of elucidate until now. And the question is, do they have the ability to do that? And I'm sure the gut reaction from many of you is the same as me, which is probably not, right? Like labeling data is very challenging. But they seem to think so. And I think it's important to remind ourselves at this time when people are shoving data into black box models that the Human Genome Project, a pretty long time ago now, thought that once we had the whole human genome, we would be able to understand the genetic base of all genetic diseases. And you'll also hear this trope, and this really irritates me and probably a lot of you too, which is people saying evolution is this optimization experiment. So we just need to harness these optimized outcomes. And of course, all of you know that there are many suboptimal or just good enough outcomes out there as well. And actually what we need to do is separate out, understand how things have evolved, separate out useful variation, and actually understand that and better contextualize that. And that's something that these approaches kind of lack. So these approaches still suffer substantially from things like taxonomic bias. Most of the data in our data sets are limited to very few species, and the kind of out there species that have been sampled are not always the best labeled. And I've worked with non-model genome assemblies, for example. We all know that the annotations of these models is not perfect, and that's a field that needs to advance as well. There's also this problem of the data quality mosaic. So when people build these data sets, it's very challenging when you have billions of sequences. Can you really throw out good sequences from bad sequences? And can you importantly stop poor quality sequences or protein structures from proliferating through your data set? And that's something that we're still not very good at. And finally, how do we define novel and useful? So each of you, when you work on your projects, novel in the context of anoles is very different to novel in the context of bacteria or fungus. And the reality is, I don't think we're at a point yet where a model can tell us what novel means in our context. It's very context dependent. And together, I think the exciting thing and the reason I'm really excited to be talking to all of you today is that evolutionary biologists have kind of a unique intrinsic understanding of these challenges specifically. Like a lot of other fields in biology use data sets or manipulate data in a way that avoids some of these challenges, whether that's just working on model organisms, whether that's working with specific protein sets. And I think many of you have a lot to contribute to these kind of agnostic searches for utility. So as far as I see it, what we ideally want is an open and scalable but agnostic discovery process. We don't want to be over-indexing on a priori disease state, for example, but we want to be able to query the tree of life and say what interesting stuff is out there. And I think there's interesting ways to tackle the challenges I already posed. So increasing our taxonomic breadth, any of you contributing to genome assembly efforts in non-model species, you are contributing to this filling in of taxonomic bias. Taking QC seriously and the increasing quality of those assemblies and annotations is also contributing to this. And then there's this question of novel and useful, and what does that mean? And that's something I'm going to talk about a little bit. And the reality is, in our case, maybe it's enough to say novel is just something useful. And that's at least kind of the working definition that I use. And that might mean finding ways to actually delineate what makes a sequence novel, what makes a protein novel. And so the ways in which I'm doing that in my work is trying to find ways to reduce the dimensionality of things like amino acid sequences here, or protein sequences and structures, I should say, sorry, and basically place these on planes. So you could just imagine reducing the dimensionality of these, high dimensionality structures or sequences, and saying which ones are more like each other, which ones are less like each other, and can we use that variance to basically identify novelty in a way. And I think one of the leaps that we're going to have is being able to sort of integrate across these different modalities in a way that we can understand and that is useful to what we're doing. So an example here might be like a sequence might be an outlier, or an individual might be an outlier if they have a amino acid sequence for a given gene, for example, that just sits way off to the side of all the other amino acids in our pool of sequences, or a protein that does something very different to a lot of the others. Similarly, we could do something like a relative outlier analysis where we identify a centroid and we say, compared to the kind of average sequence or structure, this one is very different. And so thinking about novelty in this way, I think is useful. And I'm trying to build a framework in my current work which tries to integrate across all these different data types to identify these different sort of flavors of novelty. So an example might be novelty because you have extreme structural properties. So when we decompose variation across each of these different scales, so here I've just depicted amino acid sequences. You could place individuals or sequences on this plane, protein structures, physicochemical properties, so the biochemistry of these protein structures. And then you could have, for example, just a binary protein function at the outcome. You could say, well, we know that this protein here sits at the bounds of what we've observed. That could be something like Taq polymerase, for example, if we think about physicochemical properties. Like its thermotolerance is just beyond what we observe across the norm. And we might want to label that protein or that sequence as novel in our search. Similarly, we might want to identify novelty in terms of discrete functions. So things that have similar amino acid sequences, maybe even similar protein structures and properties, but they have discrete functions. So this could be something like an enzyme where most of the protein structure stays the same, and there's just very small residue differences in the binding site of the enzyme that actually confer this very vastly different protein function. And finally, something like conservation or convergence, which again, coming back to the great talk we just had, understanding how things that are maybe different in sequence and different in protein structure can still confer similar protein functional outcomes. And a good example of this would be something like these antifreeze proteins. So we see that fish and insects both have antifreeze proteins in some species, but these antifreeze proteins have very vastly different structures, despite the fact that they have very similar functions. They stop cells from getting damaged in extreme cold. Okay, so all of you are probably thinking, well, I thought this was about speciation, so why isn't he talking about speciation? And I think one of the best ways that we can actually integrate these sort of search for novelty and across all these different indicators is to explicitly integrate speciation into these models. And the reason for that is that useful variation has explicitly involved in an evolutionary framework. The more we understand evolution, the better we can contextualize and find useful variation, because ultimately it's this backbone structure that underpins everything we observe. And the processes and sort of factors that drive adaptation and speciation are also the ones that drive useful molecular variation. So many of the differences between species are the things that we're explicitly looking for. Even things like incompatibilities, getting into like a granular level of speciation research, they underpin core differences between taxa that are important to categorize and to consider when we wonder if our data sets are full of the useful stuff, whether variation is represented well in our data sets. So this is a typical way that maybe people in biotech look at proteins and thinking about useful proteins. And I think to some extent there is this perspective that all of us have as evolutionary biologists, which is a little unique, right? We know that when you just look at a gene tree and you look at some kind of properties of a protein, that there's more to the story because of this species and gene tree discordance. So if you look at just the gene tree and if you look at just the protein space, for example, you lose a lot of the context of these different proteins, and you lack the ability to sort of follow up and interrogate these in a more robust way. And so in this case, if we add the actual evolutionary relationships between the taxa that these proteins have evolved in, suddenly you gain this kind of new perspective on the variants that you observe. And this has substantial implications, not just for better understanding these proteins, but also, as I said, following up importantly. And this critical context is something that I think far too many people right now are overlooking. And it's the reason that evolutionary studies are kind of unique in the sense that we think about the species structure and the relationship between different species, as well as the genes and the traits that evolve within those species. So I just wanted to give a few examples of why this pairing of kind of a gene view, a protein view, and an evolutionary view is really important. So say we want to identify the origin of a specific protein function or trait, right? Like this is kind of what we all do every day. If you just look at the gene tree and you look at this protein space, you can say, well, okay, proteins with this square function are each other's closest relatives when you look at sequence. They have this relatively large variance in protein space. But if you had to go out and you had to say, which other taxa could we sample to get more square-like proteins? Say square-like proteins are solving some kind of use case that we have. In the absence of this evolutionary tree, we lack the ability to say conclusively, what are the things that might correlate with this square function? And obviously, if we know that they come from both chimps and rats, for example, we know, okay, maybe mammals share square proteins. And if we want to interrogate more square proteins, like mammal lineages, undersampled mammal taxa are the ones that we should be looking at. Similarly, if we want to start to differentiate between protein function conservation or convergence, where we see functionally similar proteins in different taxa, we need the species tree to contextualize. So, if we look at just the gene tree, have all these circle proteins evolve this function independently? Have they evolved it with a common ancestor? And we can see here the amino acid sequences suggest that these two are sister to each other. This one is quite closely related, and they share very similar protein properties. But when we add the species tree, we realize that, maybe these circle proteins evolved in mammals and this algal species. And then we have to dig deeper. We have to ask the question, did the ancestor of all of these species share this circle protein? Or has it evolved convergently in these two different lineages? And I think, we intrinsically know this, but these are very different outcomes and can be leveraged very differently when we want to do follow-up of these interesting proteins. And finally, and this is the main use case for this novelty framework that I've been talking about, what happens when we observe some kind of novelty? So in this case, we have this triangle function, which has evolved, and we know that maybe it's sequence similar to these circle proteins, but it has a vastly different function when we look at its structure, when we look at the physicochemical properties. What we can do is we can say, has this novel function arisen in a single lineage with one species? Has it evolved in a very diverse taxon that it's extremely novel, like it's arisen one out of many closely related taxa. And in this case, for example, Siona has evolved this triangle protein, and we know that interrogating this branch here in the species tree is something we should do if we want to identify more triangle proteins. So it's this kind of balance of accepting that our data sets are imperfect, but the fact is we need to now direct our efforts of sampling to fill in some of these gaps that we have. And I think this speciation guided view is the way to actually do that. One of the cool things we can also do, and one of the things we do here at Arcadia, is we're doing a lot of protein design. And one of the nice things of breaking it down like this is, we can design proteins from a starting point. We can design proteins to iterate to a specific structure or a specific function. And one of the benefits of pairing these two things together is asking the question, well, if we're striving to find a protein with a given function, what's the starting point that we should use? We don't want to always be iterating from example, like a protein that's found in humans, because maybe the function we're trying to develop in this new therapeutic is actually something that's pretty close to something we see in nature. So in this case, this algae, for example, has this yellow protein here, and maybe we want to iterate to this part of the protein landscape. We know that proteins in this part of the landscape have some function we're interested in. Well, there's no point starting up here with these Siona proteins and trying to design a protein from their amino acid sequences when we can actually start with this algal sequence and we can iterate sort of in silico from there. And so being able to essentially streamline and do a more directed development of proteins is something that I think the speciation guided way of working also helps. And one of the ways we're doing that now, and something I'm really excited about, is we're actually doing sort of a full cycle test of this bioutility search framework. And the way I'm doing that is I'm using a lot of filamentous fungi proteomes. So I'm starting with 90 Aspergillus species. And I'm asking if I use all of the sequence variation we observe across these fungal species and millions of protein structures from AlphaFold, can we say something about decomposing variation to these different layers and identifying proteins that are maybe different from other proteins, proteins that are different conditioning on the evolutionary variants that we know these species have? And can we do this kind of integrative evolutionary guided discovery pipeline? And I think the nice thing with this framework is that once we develop it for a diverse clade like Aspergillus, what we're going to do is we're going to go and test it in a bunch of other test clades. And I'm sure what we'll see is that actually there are these very clade-specific variants, characteristics that we will have to tweak this model. And so I think this will be a good test of whether or not these black box models, where they hope to throw all protein sequences and structures in and kind of get something useful out the other side, are really going to work, or whether it requires this more nuanced approach. We already know, if you think of things like genome-wide repeats or things like genomic structural variation, that different clades have very distinct patterns of variants. And I think this kind of nuance is something that is going to be really important moving forward to account for. And the aim of our pipeline ultimately is to then validate some of these novel proteins or novel sequences that we find in the lab, so that we can refine this pipeline moving forward. So in conclusion, there's a lot of useful molecules out there, but finding them is challenging. And having a kind of perspective that takes into account variation across the tree of life is going to be very important for doing that. And that's a perspective that all of you and evolutionary biology's field has been trying to explain. Why are species different? What are the processes that make species different? How is that tied to their adaptive or trait variants? And I think leveraging the evolutionary structure is going to be very, very important. And the reason for that is that without this context, we're sort of in the dark, essentially. And labeling sequences in this high throughput way that other companies are trying to do, I think ignores a lot of biology that we do understand, and I think is going to be a detriment to finding interesting novelty in this kind of scalable way. And ultimately, leveraging the interesting stuff we find is also very important to consider the species tree, for example, and the speciation processes that have led to that variance. Because without those, it's very hard to do follow-up. And at a time when people are adding data, adding genome assemblies and annotations, I think that we should probably be doing a targeted effort to target lineages which are very underrepresented, that might be particularly fruitful for capturing variants we don't see across other branches in the tree of life. And I know that's something all of you, at least those of you who work on non-model and more esoteric species, are very familiar with. Is trying to think, where can we get the most bang for our buck? If we're studying very diverse systems, there are, you don't want to do another genome assembly for maybe a subpopulation of this taxon, when we could do the first genome assembly of this very understudied thing, which might, give a whole new perspective on maybe patterns of introgression across taxa, or the evolution of novel traits and things like that. So with that, I'll just say thank you. Thanks for your time and giving me the chance to talk. And if you have any questions, I'd happily take those now.
Amazing. Thank you so much, Rishi. That was amazing. So do we have any questions from the audience? Feel free to speak up or put it in the chat. No, okay. So Rishi, I have a first evolutionary biology sort of question, I guess.
Sure.
Do you include introgression as like one of these nuances in your phylogenetically informed sort of scanning for useful proteins?
Yeah, it's a really good question. So one of the things we're doing is we do like a kind of gene by gene, or orthogroup by orthogroup, I should say, breakdown of genes first when we look at like a whole proteome. And one of the steps in that process does involve looking at gene tree, species tree reconciliation. And right now, that is a coarse way of looking at that, because where we see extreme discordance for a given lineage, obviously that may represent introgression. What I would love to do is to bake introgression more intrinsically into taxa, especially taxa that we know exchange genes. And so like a good example would be like thinking about this framework for bacteria as a total nightmare, right? Because horizontal gene transfer is happening all the time. And I think it's going to be really important to account for that. And I actually don't know, for those who are doing this in a less kind of phylogenetically informed way, I guess they just ignore the fact that these things have an ancestral starting point and are being exchanged in that way. But I think you lose a lot of the nuance by doing that. But yeah, I would love to have people join us that do introgression based searches for the transfer of useful sequence. That would be great.
Thanks. Any questions, anyone? Cool. I have
no question.
Oh, sorry. We have a question from Mung in the chat. So she asks, is the process of reducing the dimensionality aware of the independence or non-independence of some regions of the protein sequences? So I guess asking about
good question. Yeah, that's a great question. Let me think. I mean, the way in which we do our sort of orthology assignment is very important. And I think there's a few that we have kind of a hierarchical approach to doing this. So we group proteins into orthogroups. One of the things we also do when we look at the dimensionality reduction is we also reduce structures down to their subdomains. So one of the challenges of using protein structures is the fact that comparing across protein domains is extremely challenging. And many of the best methods to do that are very biased by things like sequence length or the length of a given structure. I don't think there are great solutions to that right now. So there are a few methods that basically try to flatten a protein down to a kind of two-dimensional structure and then analyze it in that context. But one of the problems you get is you could have a domain that is, exactly the same as another domain, but has a big tail attached to it. And those become very hard to reconcile. I hope I'm understanding this question correctly. So I think we do account for the independence or non-independence of proteins themselves because we have this orthogroup and we have clustering and we have this gene tree, species tree reconciliation. And within structures and sequences themselves, we're also trying to account for the independence and non-independence because the subdomains and the domain structure of those proteins is important. And so I think my dream is to have a pipeline that takes into account like the sequence as a whole that contains a lot of evolutionary signal, but also then to break down the sequence and the structures into subdomains that allow us to ask questions like, where we see a kind of useful protein, where we see a change to something like a binding pocket, which is the important bit of the protein structure and the sequence that actually relates to that, because our ability to do that at a more granular level is contingent on actually finding the important bit.
Amazing. Thank you. Rishi, I have a few more questions if nobody else is going to ask. So yeah, for first one is, I guess, career related. For speciation biologists who want to move into working into this sort of amazing industry aspect, what's the best advice that you would give from like your experience?
Yeah, I think one thing that's been pretty illuminating to me is constantly finding parallels to problems that we have that are, I wouldn't say maybe solved, but have at least been tackled for often like decades, right, in evolution or even ecology. Like as we work on more things like bacterial communities, fungal communities, like I would say even like pretty old ecological theory has a place in industry. I think one of the challenges is that we're not always like well trained to contextualize our work in terms of the big picture and the bigger aims of it. And I think that is something which can be off-putting like both ways. So I think somebody who would be maybe looking to find an industry role, it's worth seeing like what are the problems being faced by industry scientists in the field that you want to work in and trying to draw the parallels yourself. So find the things that in your own work, whether it's like cancer genomics is a problem because you're looking for very low frequency, mutations or something, and you maybe work in a system where you're used to being able to separate sequencing error or, like challenging library preps and error-free library prep, whether it's something like you have a ton of experience looking at orthologs and paralogs and being able to identify gaps where people are maybe making errors to do with developing therapeutics on things they think are variants of the same gene, and maybe they're different gene copies or maybe they're paralogs, and we're looking at this differently. But yeah, the communication aspect is key. And I think there's no fast track way to doing that, but I think reaching out to people in industry, trying to maybe learn a bit more about what are the challenges they're facing, because it's not always easy to do, right? Like also we are like an exception in being very open as a company about the challenges we face. Like any of you are welcome to go and comment on any of my pubs and say, hey, you did this thing wrong and this is a stupid approach. But lots of companies don't want you knowing the stupid things they're doing. And so that is like a problem. But if you ask people, you might get a little insight into that. And so, yeah, pitching them a little bit on why your evolutionary expertise and familiarity with usually pretty bad data and challenging taxa is a use case.
Awesome. Thank you.
Thanks.
I think we might have run out of time for questions. I think we can move on to our Q&A session. So you can take it away, Parvati.
Yeah. Thanks again, Aryeh and Rishi, for very excellent and perspective shifting, I would like to talks.